So I think it's really my great pleasure to be here to talk about some of the research funding we published recently, and specifically talking about how CD8 T cell regulate the neurodegeneration. I think just some brief background. Hold on. I think the observation of the T cell in the central nervous system has been observed for quite a long time, especially when the T cells such as CD4 or CD8 T cell get infiltration to the brain. They are able to interact with different cell types such as neuron, microglia, and astrocytes. Eventually, they are able to modulate the tissue homeostasis and the pathology. And one of my research location are quite interested is the central nervous system because recent research evidence has suggested the tissue niche of the central nervous system is quite important to regulate the neurodegeneration. If we actually rule in the structure of the central nervous system, you actually see the underneath of the skull is going to be three membrane structure of the meninges, which are the dura, arachnoids, and the pili. So within the inner membrane and the intermediate membrane of the meninges is the CSF. And eventually they actually reach the parenchyma. So we know that the peripheral immune system can get access to the meninges through the blood circulation. And eventually they will cross the inner membrane of the meninges and reach the parenchyma. But in many cases, especially in the brain injury or neurodegeneration disease, the structure of the meninges is actually compromised, which leads to more accumulation of the immune system in the parenchyma. So taking the example of the Alzheimer disease as example, in the parenchyma region, they have very specific accumulation of the protein aggregates, such as A beta. So this A beta actually can prime the homeostatic microglia to become this dysfunctional state of the microglia, especially by production of the cytokines such as TNF-alpha and A1 beta. But in addition to that, the A beta also creates this neuroinflammatory environment, which potentially can regulate the peripheral immunity. So the biology question we would like to specifically address for this project, and some open question still remain in the field, is how the tissue niche or the disease environment can modulate the peripheral immune system response, and how they actually eventually control the disease progression. So to address this question, we actually use a very established mouse model, these 5SD mice. So these mice actually carry 5 genetic mutation, and they can fully recapture the pathology, including the A beta plaque deposition and the microglia activation, and some of the neuronal loss in the hippocampus and the cortical layer 5, and eventually this mouse model also developed some memory function deficits. So what we did is to perform this single cell RNA-seq from the immune cell, isolated different tissue location, the parenchyma versus the meninges. So in the parenchyma, we actually saw the microglia immune cell and also the non-microglia immune cell, and mix at a ratio of 1:1. So the purpose is really to boost the resolution of the immune cells from the peripheral system. In addition, we also use the immune cells from the meninges as a comparison to really check the difference of the alteration of the immune system in this two tissue location. So we did both RNA-seq and T-cell laboratory analysis. So the first data analysis we actually get from this single cell RNA-seq is to check the proportion of the immune cluster in the non-TG versus disease 5SD mice, specifically in the brain. So what we see is from this bar graph is that the accumulation of this class 12 disease-associated microglia only present in the 5SD mice compared to the non-TG. But in addition to that, we also see another cluster 1, which are the CD8 T cell, further upregulated in the 5SD mice compared to the non-TG. But where other immune cluster we identify in this brain environment, we don't see any increase or even reduction. which implicate that maybe the accumulation of CD8 T cell somehow involved in the progression of the disease. So to further validate with this result, we actually performed some of the flow cytometry looking at CD4 versus CD8 T cell. So from this flow plot as well as statistic, we see it's quite a specific accumulation of CD8 T cell in the 5-ICD mice brain, where we don't see any CD4 further accumulated in the disease mice brain. And this accumulation is quite tissue specific because we examine other tissues such as meninges and the liver and the lung. We don't see any accumulation of CD8 T cell present in this tissue. I think the following question for us is really to understand, which signal actually mediate the recruitment of CD8 T cell to the 5-ICD mouse brain. So to further address this question, we actually run some of the bioinformatic analysis, especially focus on the expression of the ligand receptor expression. So we eventually identify a very interesting and unique chemical ligand receptor, CXR16 and CXR6. So from this UMAP, looking at the expression pattern of these two genes, we definitely see this chemical ligand CXR16 MR level are really highly and abundant expressed on the microglia, which labeled by this dash circle. In addition to that, the corresponding chemical ligand receptor CXR6 is also very selective, abundant expressed on the T cell, including the CD8 T cell. So using the flow cytometry, we eventually demonstrate this chemical ligand is quite a selective expressed on the microglia compared to the other immune cell or non-immune cells. In addition to that, we also see the further upregulation of this chemical ligand on the 5-ICD mice brain. And similarly, when we check the CD8 T cell, we see the brain CD8 T cell actually further upregulate the chemical receptor expression. And this expression can be further upregulated in the disease environment. And this phenotype leads to more accumulation of CXR6 positive CD8 T cell, specifically in the 5-ICD mice brain. But another interesting observation we do see is the CD4 phenotype, because we know the CD4 also express the CXR6, but we don't see any disease-dependent regulation of the chemical receptor on the CD4 T cell, which implicate the regulation of the chemical receptor is quite specific to the CD8 T cell in this disease environment. So moving forward, I think as the immunology, we are quite interested is, how this CD8 T cell perform some immunological features, because when the T cell get into the brain, they are going to meet different type of antigen as well as some chemokine or cytokine production, which can lead to the clonal expansion of the T cell. Eventually, once the retraction, they can become some homogeneous population. And from the literature, previous literature, some of the evidence has been suggest that the clonal expansion of CD8 T cell indeed present in the disease environment, especially this 2020 paper from David Gates identified the clonal expansion of CD8 T cell present in the CSF of the AD patients. In addition to that, another research group also identified the clonal expansion of CD8 T cell in the aging people and then mouse environment. But because in the patients or human, it's really hard for us to really understand in the different tissue environment, especially looking at the meninges versus parenchyma. I think for us, we can actually take advantage of the mouse model, especially compare the antigen T cell response in this meninge and the parenchyma region. So to do that, we decided to first looking at the antigen T cell response, focus on the T cell repertoire data analysis. Especially one of the readout we can check is to group the clonal expansion of the CD8 T cell based on the clonal type size. So by running this bioinformatic data analysis, we first use this pie chart to group the proportion of the T cell response by compare the tissue location. So from this pie chart, we do see that majority of the clonal expansion of CD8 T cell are only present in the brain compared to the meninge. You can see the large fraction of the large and the medium clonal type size are actually present in the brain, while in the meninge, there's very limited clonal expansion of CD8 T cell. In addition to that, we're also quite interested what's the difference of the clonal expansion of CD8 T cell in the control versus disease mice. So from this pie chart plot data analysis, we do see the clonal expansion of CD8 T cell are further upregulated in the disease environment. So taking together, this data actually suggests in the AD disease environment, they have some tissue, both have tissue and disease effect in regulation of the antigen T cell response. But because this is only the bioinformatics data analysis, for us, we really want to identify some of the surface marker to indicate the clonal expanded of CD8 T cell. In that case, we can study their function. So to address this question, we first further look into some bioinformatics data correlation analysis, especially focus on this percent correlation, looking at the clonal expansion of CD8 T cell versus the gene expression. So one of the top hits we actually identify is this gene, the PD-CD1. So the PD-CD1 actually encodes the PD-1 expression. So this data actually maybe suggests that the clonal expanded CD8 T cell could be the PD-1 positive CD8 T cell. So to test this hypothesis, we first further looking at the immunohistochemistry staining of the CD8 T cell from the both 5IC mice as well as 80 patient slide. So from this imaging staining, we can appreciate that the CD8 T cell indeed co-label with PD-1 expression, especially labeled by the red color as well as the green color. It's also very interesting that these PD-1 positive CD8 T cells are quite close, are really like spatial localized with A beta. So I think now coming to this end, I think for us now we identify the tissue and the disease dependent clonal expanded CD8 T cell specific in the 5IC mouse brain. For us, we really want to know what exactly these clonal expanded CD8 T cells do in the disease environment. So to further address this question, we decided to first focus on the microglia response because the previous data showing the chemical ligand receptor mediated crosstalk between the microglia and the CD8 T cell. So we first isolate the microglia from the 5IC T cell for knockout versus the 5ICD mouse alone, as well as the microglia from the B2M knockout versus the 5IC mice. So the purpose is really to check the microglia response in the absence of the T cell or in the absence of the CD8 T cell response. So first we did some of the gene set enrichment data analysis and separate the microglia into the homeostatic microglia subset M0 versus the disease-associated microglia. So by comparing the T cell knockout versus wild type, we do see that in the absence of the T cell or in the absence of CD8 T cell, the microglia, the pro-inflammatory response signature are further upregulated in this knockout microglia cell, which implicates that without the T cell or without CD8 T cell, their pro-inflammatory response are further increased. Which again support maybe the CD8 T cell in the disease environment actually perform some regulatory function. So the concept of the regulatory biology is actually present maybe 20 years ago. The people actually identified some of the subsets of CD8 T cell can upregulate some changing factors such as Helios, and to program the regulatory function and control the TFH response and suppression of the autoantibody response. But later on, the people from Stanford, the Mark Davis group, also identified there are some of the antigen-specific CD8 T cells somehow can inhibit the more specific CD4 response in the EAE mouse model. So they also define this CD8 T cell regulatory, the suppressive function. So to further test our hypothesis whether these CD8 T cells are actually perform the regulatory function, we actually accumulate the gene signature from their data set and apply to our data, especially compare the clonal expanded versus non-clonal expanded CD8 T cell or PD-CD1 versus PD-CD1 negative cell. So from the GSA plot, we can appreciate that this regulatory CD8 T cell signature are significantly enriched in the clonal expanded PD-CD1 positive cell, which again support our initial hypothesis these CD8 T cells indeed have some suppressive function. So to further demonstrate this angle, we actually adopted some in vitro co-culture system. So we actually saw this CD44 high CD8 T cell from the spleen, and then give the cytokine stimulation for 2 days, and we eventually separate the PD-1 positive CD8 T cell from the PD-1 negative cell and co-culture with microglia and give stimulation to induce the cytokine production on the microglia. So from the flow cytometry, first we can appreciate that compared to the unstimulated microglia group alone, the stimulated microglia actually significantly upregulate the cytokine production, including TNF-alpha and L1 beta. And it's quite interesting that when we actually culture the PD-1 positive cell with the microglia, and we can see the cytokine production of the microglia are significantly reduced, whereas such inhibitory effect on the PD-1 negative cell is actually absent. So which implicate indeed this PD-1 positive CD8 T cell have some regulatory function. And to further demonstrate this data is true in the brain CD8 T cell, we also co-culture the microglia with the brain isolated CD8 T cell, and we also see some inhibitory effect on the cytokine production in the microglia when we co-culture with brain CD8 T cell. So taken together from the data I have been present, we actually identify the tissue and disease-dependent accumulation of CD8 T cells specific in the 5SD mouse brain. And then we also identify this CD8 T cell somehow can crosstalk and communicate with microglia, and potentially they upregulate the chemokine receptor CXCR6 and talk with the CXCR16 positive microglia. And this CXCR6 positive CD8 T cell will undergo the clonal expansion, and which is characterized by the PD-1 upregulation. And eventually, this clonal expanded PD-1 positive CD8 T cell actually perform the suppressive function and to inhibit the pro-inflammatory activity of the microglia and slow down the disease progress. I think another point I would like to mention is that there are another human study from the David Gate group in the Northwestern group. They also identified the clonal expanded CXCR6 positive CD8 T cell present in the CSF of the AD patients, and potentially this CXCR6 positive CD8 T cell in the CSF are able to interact with CXCR16 positive monocytes. Again, this human study somehow gives some additional support to our mouse study. I think beyond that, I would like to extend the discussion about this regulatory CD8 T cell function in the field. I know the regulatory function of CD8 T cell is not that well established, but overall, I think based on the publication from recent paper, we do appreciate that in addition to our funding, this regulatory function can be identified in different disease environments, especially the Mark Davis group identified this regulatory CD8 T cell from the virus patients, the COVID patients. In addition, they also identified the regulatory CD8 T cell present in the autoimmune, the EAE mouse model, as well as recent research group from the Pittsburgh. They identify even in the exhaustion T cell state, the CD8 T cells are still able to perform the suppressive function and somehow implicate the disease environment indeed can allow the CD8 T cell acquire the regulatory T cell biology. But in addition to that, we also have to appreciate the pro-inflammatory function of CD8 T cell especially in the neurodegeneration field, because a lot of people actually identify this pro-inflammatory function of CD8 T cell in different disease models, especially tau pathology, also this 3D human co-culture system, as well as the aging-induced neurodegeneration disease mouse model. But I think again, all this data combined together implicate maybe the complexity of the CD8 T cell response in overall the disease environment. This is maybe the factor we need to consider in future for us to fully understand how the T cell can evolve in the context of the neurodegeneration. But maybe moving forward, I think another some of the research funding we can appreciate is that some of the implication is indeed present looking at the MHC complex allele, especially HAL analysis. These two findings indeed support there are some association of the protective T cell response with neurodegeneration, especially Alzheimer's disease and Parkinson's disease. But in addition, they also report some of the detrimental T cell response actually associated with the accumulation of the AD pathology. Indeed, which actually gives us some maybe further interest to study how the T cell response can evolve in the disease environment. Also, think about the progression of the disease stage, which could be another factor we need to consider in future. But I think last but not least, I think I would like to extend my acknowledgement to this lovely lab during my academia journey. Especially, I would like to first thank my PhD advisor, Dr. Hongbo Chi, for his tremendous support as I'm a graduate student in the lab. And I think the training I really received in his lab is really rigorous. And I extend all of my interest to the neuroimmunology field. And also, in addition, I would like to extend my acknowledgement to my current postdoc advisor, Dr. Thomas Jude. So he allowed me, give me some freedom and also flexibility to combine my research interest in immunology to the neuroscience field. So I think without any hesitation, I think I would like to stop here and take any questions you have. Yeah, thank you.